Hello, back again. The routine use of genetic and genomic sequencing in the treatment of cancer has been steadily rising over the past year. Also, an increase in the availability of biomarker-based clinical trials. That leads to the question how to explore this next-gen sequencing and how it impacts the transformation of the cancer treatment. Let's look at the case in the Avera Cancer Institute. Today, lead clinician bioinformatics analyst Priya Swaminathan is going to present you CancerTrialMatch. Priya, the floor is yours.
Hi, everyone. Thank you for your kind introduction, and thank you to the organizers for this opportunity. It's my pleasure to introduce our web application for the curation and matching of clinical trials at our Precision Oncology Center. The outline of my talk is going to be first introducing precision oncology or precision medicine, talk about our Precision Oncology Center, introducing clinical trial, where you can find this information, and the goals of this web application and the workflow, and then the demo of the 3 interfaces: curate, update, and browse. So what is precision oncology or precision medicine? It's giving the right cancer treatment to the right patient at the right dose at the right time. So on your right, what you see here is the UC Davis precision medicine model. So here you see the intersection of the patients, cancer patients, biomarkers, environmental changes, and then the behavioral changes all put together. And our Precision Oncology Center called Avera Cancer Institute is in South Dakota. It serves cancer patients for prevention, diagnosis, and treatment. Roughly about 3,500 newly diagnosed patients per year across 5-state region. For all cancer types, we have 2 programs, cancer research and cancer genomics, and molecular tumor board happens every week. What is a clinical trial? It's an experimental study. It can be of any phase. Then duration could be from a month to several months, and number of participants could be different and for a different purpose, and different side effects. So you can find clinical trial information by going to this API, clinicaltrials.gov. What you can see is starting from which disease it's addressing and what is the intervention or the treatment, which phase, what is the eligibility criteria, and the title, description, and so on. So the goals of this web application, before we started this developing this application, when we are looking out for open source, there is one that's available, but it is vast and not easily customizable for our needs. So hence, with the intention of a no setup, easy to use interface, and also with features of interest, we started developing this. And because of this, with also the increase in the number of clinical trials over time, and this application would be apt to manage and access trials easily. Hence, increase in the clinical trial enrollment, and also be able to find and explore suitable clinical trial options for every patient. So the workflow of this web application, what you see here are the different interfaces. So the curate interface, edit, and the match interfaces. So the curate interface is a semi-automated interface. So it's where you curate information partially by manual curation, and then the rest captured from the API clinicaltrials.gov, saved as JSON format back to the Mongo database. And then anytime you have changes to be made for any clinical trial, you can read from the Mongo database and then make the changes and save it back again to the Mongo database. So here is the match component, where you can basically match trials to patients and patients to trials as well. So when you talk about patients, we have clinical data, demographic data, and genomic data available. At this point, we have the genomic data available in the SQL Server database. And this is what I meant by the patient-centric and the trial-centric approach. So when you say patient-centric, as I said, we have the genomic testing results plus their clinical and demographic data, which you can use to match to clinical trials. Similarly, clinical trials with a certain set of eligibility criteria could be matched against patients. So what you see here is the OncoTree, and this is what we use during our curation process for the ontology classification for disease type. So you have different levels of information starting from level 1 to level 7, level 1 being the tissue site. And this is the JSON format after we finish the curation. And this is how it is saved back to the Mongo database. And this is the text-based human readable format. And what you're about to see here is a demo of the 3 interfaces: curate, update, and browse. Stay tuned.
Let's see how to curate a clinical trial. So in this page, you see different tabs for entering different information for any clinical trial. First is the NCT ID, which holds the basic details for any clinical trial, and disease information, biomarker, documents for the reference, and finally, summary page to review and save. Let's start. This is the NCT identifier, and then the protocol number, and then trials offered by which vendor, and then the name of the trial and disease category. This being a lung trial, choose medonc lung. And then the principal investigator information. Currently, I don't have any, so I choose not available and search. And this is where it interacts with the clinicaltrials.gov API and brings information like title, summary, sponsor, phase, age, conditions, and so on. So move to disease. So where you can enter the overall status for this trial, and then the disease summary, which is non-small cell lung cancer, and choose if it is include or exclude criteria. So I have chosen include. And then this is where we enter the ontology classification from OncoTree. And there are 7 different levels of information. So for lung, we choose the tissue site as lung, and non-small cell lung cancer, and advanced stage metastatic. This is the stage information for the disease, and click on add. Shows this table with the disease and the stage, and move to biomarker. Here we have 2 different tables, one for the line of therapy, arm status, and the other one is for adding the biomarker. So you could basically enter information in 2 different ways, and these are the different cohorts. So you can enter information for each of these cohorts separately using this green icon plus, and otherwise you can choose to click on this green icon that says add common LOT and arm status for entering common information for all the arms at the same time. So we can do this same for the biomarker section as well. So clicking on this, enter the line of therapy information and add, and shows this table with all the information for the line of therapy and arm status, and selecting this icon, and you can choose between include or exclude, and the gene information, looking for EGFR mutation, and if you have any information about the variant, function, and gene too, then you can enter those. Otherwise, you can leave it as not available, and add shows the new table with all the information that you selected, and move to documents, and this is the place where you enter information about the reference document, and then the date when it was created or updated, and then the location of the clinical trial where it's available, and then move to browser, and this is the summary page. You can review all the information, and you can see the part where you curated manually, and then the part where you uh, captured from the clinicaltrials.gov API, and then the line of therapy and biomarker information, and also about the disease and the stage information. So everything looks fine. So clicking on confirm will save all these information in JSON format to the Mongo database in the backend.
To update an existing clinical trial, we use this interface. On your left, you see a sidebar with a list of existing clinical trials, and the main panel has part 1 and part 2, which holds all the information for any selected trial. Let's say we would like to update this clinical trial, and once you select this clinical trial, you click on edit, and this opens up the information for this particular trial in the main panel. And I have a total of 2 changes to be made. One is the hold status, another one is on status. So in the first table, I have the hold status, and this has to be changed from open to closed. Once changed, click on Save Verify and move on to the next table to see if you have any more changes here. If not, click on Save Verify. Likewise, look at the different information, and then we move on to part 2. And here we have the arm hold status in this table. Here, the first arm, the information needs to be changed from open to closed. Once done, click on Save Verify and check the next table, and if there are no more changes, click on Save Verify. Once all the changes are made and checked, click on Submit, and this would be the final step. Once submitted, these changes are saved back to the Mongo database. This interface is designed for browsing or searching clinical trials. What you see here in this page are the different columns containing different information for any clinical trial. You have searches possible using this global search bar and for each of these columns. And there is an arrow here that you can click and open to look for more information for any clinical trial. And then clicking back brings it to the collapse format. And let's say I would like to use this global search bar to search for lung trials. And these are the results showing in the front display. This actually searches the complete table. And you can also make searches using these search bar for every column. Let's say I'm looking for just-in-time trials addressing solid tumors, and this would be the search results. And clicking on this icon takes you to a sidebar where you have inputs for selecting trials based on a set of criteria. And there are options where you can look at the closed trials only instead of the open trials, which is now what is showing in the front display. And then you have the reset trials and the collapse all options. Once you finish selecting any trials to look at, you can reset back, and then collapse all helps you to collapse the information after you have looked at the expandable format. Let's say I would like to look at all the closed trials. In this case, this is how it looks. And reset trials brings it back to the original display. And let's say I don't want to look at all the different columns which is showing in the front display now. Instead, I would only want to look at columns selectively. In that case, I can go ahead, select all those, and that will be shown in the front display. And reset trials brings it back to the original display. And moving on, let's say I would like to browse or search for clinical trials with advanced stage non-small cell lung cancer. In this case, I choose these two inputs and click on filter, shows the results for the open trials. If you want to look at the closed trials, you can click on this checkbox to see the closed trials. And reset brings it back. And this is how we use the different search bars and also our select inputs for different searches and other features.
Welcome back. Thank you all for listening. And this is our lab, and thank you for having me.
Thank you so much. We have a space for one question. So I'll start with, are you hosting the MongoDB database on a cloud service like AWS? Maybe you have some challenges linking the database connection to your Shiny app backend? Priyam? Hello, hello, hello. There is a question. Okay, so I guess that we can answer it in the comments. Thank you so much for that session and for your talk, Priya. Thank you so much.